Alright, section 3.2, we're going to first go through part of the study guide and then stop and go through the material a little bit more. So starting out, signal transduction and cell-to-cell signaling. Cells are constantly responding to external and internal stimuli. Each cell in a multicellular organism is an independent agent. This is important to note that if an individual cell, even in its differentiated status, is taken out of the tissue and put into an artificial environment, its phenotype will change. It is a bit constricted on how far its phenotype can change because of its differentiated status, but its phenotype will change. So each cell in a tissue in an organism is an individual agent that is responding to the environment that it is in. And the main thing to note about that, there's not some sort of central controller controlling each individual cell. Okay? A cell's behavior, phenotype, is dependent on the environment it is in. A cell's phenotype will change if it's removed from its natural environment. Its behavior is dictated by molecules, also called factors, in a cell's extracellular environment, and whether a cell has the molecular machinery to act on these factors and formulate a response. And B is just noting that a cell is stuck in its differentiated status where certain genes are not available to it because they've been silenced. So if it does not have the factors, the necessary molecules, the proteins, to respond to an external stimuli or an internal stimuli, it will not respond to those cues. Example, a neuron lacking acetylcholine receptors will not respond to acetylcholine in its synaptic cleft. You can have all the acetylcholine in the synaptic cleft possible, but if those acetylcholine receptors are not available, then the cell will not respond to the acetylcholine. The mechanism by which cells respond to internal and external factors is called signal transduction. And we've already had an example of signal transduction. We just didn't call it by that name, and that's neuronal signaling. Cells communicate with each other through secreted factors such as hormones and neurotransmitters. The figure below shows the four modalities of cell-to-cell communication. So we have a picture here of the four modalities. Contact-dependent signaling. So contact-dependent signaling, the signaling molecule is actually a membrane protein that is tethered to the membrane, and once the two cells become close to each other, then the receptor will bind to the signaling molecule and you will get some sort of response by the target cell. In the case of paracrine signaling, in a tissue, a signaling cell will secrete out signaling molecules, which are being shown with the red dots, and those will go ahead and diffuse and bind to receptors on the target cells, and that will lead to a response by the target cells. In synaptic, we've already gone over this. You have a synaptic connection, neurotransmitters are released by the presynaptic cell, and they will diffuse into the synaptic cleft and bind to receptors, transmediated channels, etc. on the postsynaptic membrane, and that will lead to a response by the target cell. Endocrine signaling is kind of like paracrine signaling, but it's more long distance, where the endocrine cells, the cells that are secreting the signaling molecule, are far away from the tissue that they're targeting. The signaling molecules must enter the circulatory system, and they will then get to the target cells and bind to the correct receptors, and that will lead to a response by the target cells. In the case of cell-to-cell signaling, the signaling cell is one which releases the signaling molecule, and the target cell is expressing the receptor for the signaling molecule. It is important to note that if a cell is not expressing the receptor, then it is deaf to that signaling molecule. The binding of the signaling molecule to its receptor on the target cell leads to the receptor's activation. The activation of the receptor leads to a molecular pathway within the target cell to be turned on, which eventually leads to the activation of an effector protein. The activated effector protein formulates the correct response by the target cell to the initial signaling molecule. Now we have a picture here. It's a very generic picture. Let me just say that right out. Where we have a signaling molecule binding to a receptor, and this receptor is on the cell membrane in this case, and that activates the receptor. The receptor will then go on to activate some intracellular molecules. And that will eventually lead to possibly different types of responses. It could be a change in cytoskeleton protein, transcription regulatory proteins might be activated, or metabolic enzymes. And this can lead to altered metabolism, altered gene expression, altered cell shape or movement. We've already seen an example of altered shape or movement when acetylcholine binds to the acetylcholine receptors on a myocyte. That leads to calcium being released in the cytosol, and that leads to muscle contraction, which is because cytoskeleton proteins are going to change their shape. Note the following. A cell can only respond to signaling molecules for which it has receptors. Even if different cell types within a multicellular organism are expressing receptors which bind the signaling molecule, the signaling pathway activated within could be different, which leads to different responses to the same signaling molecule by different cell types. So we have an example here of acetylcholine as the signaling molecule. In hard pacemaker cells, acetylcholine binding to an acetylcholine receptor will decrease the rate of firing, of action potentials being fired. In the case of the salivary gland, when acetylcholine binds to the acetylcholine receptor on the salivary glands, you have the secretion of saliva. In skeletal muscle cells, as we saw in section 2, when acetylcholine binds to the acetylcholine receptors, you have muscle contraction. Completely different responses, but the signaling molecule is exactly the same. And why is this the case? Well, this goes back to differentiation. Even though these cells in a single organism will be genotypically equivalent, because of their differentiated status, their response to the signaling molecule will be completely different. Even though the cells pictured above are genotypically equivalent, each one is expressing different signal transduction pathways for acetylcholine, which leads to a unique response for that cell to acetylcholine. A signaling molecule will be ignored by cells lacking a receptor for it, or the internal pathway necessary to formulate a response. Thus, through the process of differentiation, cells in different tissue acquire unique phenotypes even though genetically equivalent. For some cellular responses, a cell must receive multiple signals. Some responses require activation of multiple signal transduction signaling pathways. They include survival, inhibition of apoptosis. This is very important. You don't activate apoptosis. Apoptosis is actively being inhibited by cells that are alive. The lack of survival signals will lead to that inhibition being removed from apoptosis, and therefore apoptosis will be triggered. Growth and division, progression through the cell cycle. Differentiation. Cell differentiation requires the absence of growth signals and the presence of survival and differentiation signals. A cell will not differentiate if it's actively dividing. And of course, in this case, you'll also need the survival signals, which will inhibit apoptosis. Cell growth and division requires the absence of differentiation signals and the presence of survival and growth signals. The absence of survival signals will lead to programmed cell death, apoptosis. And this picture is just showing you that, where A, B, and C are survival signals, and that tells the cell to survive. In the case of growth and divide, you have the survival signals A, B, C, but you have these two other signals coming into the cell, D and E, which tell the cell to go ahead and divide, or pushes the cell through the cell cycle. In the case of differentiation, you have the survival signals A, B, and C, and now you have two different signals, F and G, which will push the cell to differentiate into a specialized cell type. And in the case of the absence of A, B, and C, the survival signals, well, the cell will then go through apoptosis, programmed cell death. The signal transduction pathway. We will only discuss signaling pathways whose receptors are embedded in the cell membrane. There are many signaling pathways in which the receptor is intracellular, but the biology is the same. So there are three types of receptors. We have ion channel coupled receptors, and these are the ones that we talked about in section 2, transmitter-gated ion channels. We have G-protein coupled receptors. And G-protein coupled receptors, once activated, for example right here, they go on to activate a protein called the G protein. In this case, in the first panel, the G protein is inactive, but then it becomes activated here. And that activation, because of its ability to bind to an activated receptor, leads to certain changes that we'll be discussing shortly, and that leads to the activation of a signal transduction pathway. Enzyme-coupled receptors. In this case, what you have is monomers of the receptor which dimerize once the signaling molecule is bound, and that leads to the activation of the receptor. And again, G-protein coupled receptors and enzyme coupled receptors are the topics for the rest of the section. So we'll be discussing those in more detail shortly. Ion channel coupled receptors. These are the ligand-gated channels, i.e. transmitter-gated channels, discussed in the previous section. We will not discuss these receptors in this section. G-protein coupled receptors, GPRCs. All GPRCs are composed of single polypeptide chain which passes through the membrane 7 times. Upon binding its signaling molecule, a G-protein coupled receptor is activated. It will then activate a G-protein within the cell. It's very important to note that this is a class of proteins. There's many different types of G-protein coupled receptors, but they all have something in common, and that is that it's a single polypeptide chain that passes through the membrane 7 times. Enzyme-coupled receptors. Enzyme-coupled receptors are monomers, which have an extracellular binding pocket for its signaling molecule. The intracellular receptor tail either has kinase activity or a protein kinase bound to it. Note, kinases are enzymes which will phosphorylate their substrate using an ATP. So let's take a look at that. So the substrate A is phosphorylated using an ATP, giving you A phosphorylated. The enzyme for this reaction is a kinase, because as we said in section 1, every reaction, every chemical reaction in a cell has an enzyme associated with it. The opposite reaction of A phosphorylated going to A plus an inorganic phosphate is catalyzed by enzymes called phosphatases. These are 2 different enzymes, 2 different reactions. It is being catalyzed, the phosphorylation, by a kinase, whereas the phosphate group is being removed by a phosphatase. Again, these are classes of enzymes. Okay? Enzyme-coupled receptors are activated through dimerization. Again, the picture here was showing you that they're monomers, right here, and upon binding the signaling molecule, they dimerize. That dimerization leads to their activation. So you can essentially say that the quaternary structure changes upon binding of the signaling molecule. Whereas before they were monomers, but once the signaling molecule is bound, they become dimers. Now, important to signal transduction is molecular switches. There are 2 types of molecular switches. There's signaling by phosphorylation, being shown right here, and there's signaling by GTP binding, right here. Now, in the case of phosphorylation, we just kind of talked about it. Whereas maybe the protein is in the off state when it's not phosphorylated, which is the example here, and upon phosphorylation, it is activated. And when it's dephosphorylated by a phosphatase, it is inactivated. Okay? In the case of signaling by phosphorylation, phosphorylation could be the off switch, whereas dephosphorylation, meaning the protein is not phosphorylated, is actually the on switch. But in the case of the GTP binding proteins, which is being shown in this picture here, when the protein has GDP bound, it is inactive. And when it has GTP bound, it is active. So the activation step is being shown right here. Signal comes in. The GDP is switched out for GTP. This is just the switching process of one molecule for another. Okay? You're not actually phosphorylating the GDP. And then the off switch is when the GTP is hydrolyzed back to GDP and the protein moves to the off position. a phosphorylation switch where the protein is active when phosphorylated and inactive when it is not, or the protein is active when it is unphosphorylated and inactive when phosphorylated. Phosphorylation of these types of molecular switches is catalyzed by protein kinases, and dephosphorylation is catalyzed by protein phosphatases. Monomeric GTPases, or GTP binding proteins, these are equivalent terms, is active when they have GTP bound and inactive when they're binding GDP. Monomeric GTPases have GTPase activity. So this is important. The protein that is the switch, it itself has GTPase activity, meaning that when it has GTP bound, it can actually dephosphorylate it to GDP. Keep that in mind. In cells, the concentration of GTP is higher than GDP, thus at relatively slow rate, inactive monomeric GTPases will switch out the GDP for GTP, but the monomeric GTPases have GTPase activity and will hydrolyze the bound GTP to GDP at a similar rate and inactivate themselves. So we have our monomeric GTPase. And it's in the off position when GDP is bound. Now, the same protein, when it has GTP bound, is in the on position. What we mean by on and off is mean that when it's in the on position, it can go on to do something. Where it's in the off position, it cannot do what it's supposed to do. Okay? Or what it can do when it's in the on position. So, this protein, this monomeric GTPase, has GTPase activity. And what does a GTPase do? A GTPase will take GDP and hydrolyze it to GDP. This is the enzyme that is involved in this reaction, because this is a chemical reaction. As we said before, every chemical reaction in the cell has an enzyme associated with it. Now, since it has GTPase activity, this process of turning off is sort of active all the time. And since there's more GTP in the cell than GDP, then just through the process of binding and unbinding and weak interactions, what you have is this process happening kind of by itself, where the GDP is kicked out and the GTP replaces it. Okay? A protein called guanine exchange factor, GEF, substantially increases the rate by which an inactive monomeric GTPase exchanges GDP for GTP. This rate of exchange far outpaces the rate by which a monomeric GTPase's GTPase activity can hydrolyze GTP to GDP, thus increasing the concentration of activated GTP-bound monomeric GTPases. So when we're looking at this picture, this process by itself, with no other external things happening, where GDP is kicked out and GTP replaces it, and then that GTP is hydrolyzed, these are two competing processes that are basically happening almost at equal rates. So the concentration of the on protein, the GTP-bound protein, does not increase if nothing else happens. Okay? So, what you have is another protein called a GEF. This is a completely another protein, and what this will do is will increase the rate by which this happens. Okay? And if you increase the rate by which GDP is switched out for GTP, then this form of the protein increases dramatically. Okay? The form that is in the on position. Just because now the rate by which you're activating the protein, switching out GDP for GTP, is outpacing the rate by which the monomeric GTPase can hydrolyze that bound GTP. So the concentration of the monomeric GTPase or GTP binding protein with GTP bound will increase. A protein called a GTPase activating protein, GAP, substantially increases the GTPase activity of monomeric GTPases. Therefore, GAP binding will lead to quick inactivation of monomeric GTPases by increasing the rate of GTP hydrolyzation to GDP. So what is a GAP doing? A GAP will bind to the monomeric GTPase and increase its activity, so now this hydrolyzation reaction is happening at a faster rate. And this is what the bold arrow is supposed to be showing you. Okay? So in the picture above here, if you activate a GAP, which is a protein by itself, so we're going to put a little square around it to denote that it's a protein itself, what it does, it increases the rate of this thing happening. Okay? Now you might be saying, well, that doesn't really solve the problem because you have a GEF that is active on the other side. But what will happen is when a GAP is activated, this signal will eventually disappear. And once that disappears, then this sort of disappears too and goes back to its normal state. But now the rate by which the hydrolyzation process is occurring will increase dramatically, and this form of the protein will start to dominate. Meaning you're going to have a higher concentration of the GTP binding protein with GDP. Second messengers are small organic molecules or ions. Examples of second messengers are calcium, cyclic AMP, cyclic GMP, IP3, diacylglycerol or DAG. We've already seen an example of calcium, and we know that if you increase calcium concentrations within the cytosol of a cell, the cell will respond in some way. Now, all these other molecules are just like calcium. You increase the concentration of these molecules within the cytosol of the cell, and there'll be a response by the cell. So these are all concentration dependent, meaning the activation of these molecules is simply means that you're increasing the concentration of these molecules in the cytosol. And there are active processes that are always on that are working to keep the concentrations of these things at a very low level. And the activation process being that you are turning them on, increasing their concentration, is dependent on the correct signal transduction pathway being activated. So for the case of neurons, we saw that an action potential will lead to depolarization of the presynaptic membrane, and that will lead to voltage-gated calcium channels being activated. And when we say voltage-gated calcium channels are being activated, we just mean that they open. And that leads to an increase in calcium concentration. Right? And that's what we mean by activation of calcium. So even without these brackets, when we have this activation step, what we mean is that the cytosolic concentration of calcium increases. And of course, once that happens in the axonic bulb, you have neurotransmitter release into the synaptic cleft. And this is a signal transduction pathway. Now, we also know that the concentration of calcium within the cytosol is always being lowered by processes that are always active, and that is the calcium ATPase, which is moving calcium out of the cytosol into the ER, and we also had a second one, which was the sodium-driven calcium exchanger, which is a cotransporter that moves calcium out while bringing sodium in, an antiporter. So those processes that are keeping calcium levels low in the cytosol are always active. It's just when voltage-gated calcium channels open up, the rate by which calcium is coming into the cell is much higher than the removal process, so the concentration of calcium spikes within the cytosol. Activation of subsignaling molecules will lead to a dramatic increase in a second messenger cytosolic concentration. This increase will lead to a response by the cell. Example, increasing calcium concentration in myocytes will lead to contraction. Okay, and we'll stop here and continue with the rest of the section in the next video. Thank you.